Okay. Good evening, everyone. Today's topic is Drosophila Developmental Biology Basics. Before starting to this topic, I want to tell that the developmental biology of Drosophila shares a very large part of whole developmental biology syllabus. So, as this part is very lengthy and there are there are a number of genes and proteins involved in this process, so I have tried to make this topic easier to understand or remember. But I want all you people to be with me so that we both can learn this Drosophila developmental biology process in a better way. So let's start with the introduction part of Drosophila. First thing is that Drosophila is a model organism, as we all know. We all are aware of this Drosophila. So Drosophila is basically a very popular model organism that is commonly known as fruit flies. Then it is it is widely used in biological research and have made a very significant contributions to the greater scientific community over the last century. So why it is the model organism or why it is popular? The question arises. So for that, the reason is it is very easy to culture them in mass. Part of this, they are very inexpensive to maintain in the laboratory and have a very simplified genetics. So why it is easy to culture them in mass? Because their generation time is very short. And to study any type of disease or any function of a gene or anything in this biology, so for that we need a mutant organism. So Drosophila is very easy to mutate because they have very small generation time, and the result of that experiment is also we can get in less time because it's easy to culture Drosophila. A part of this, the generation time is also very short. So what will be the topics that I will be covering in this whole Drosophila developmental part? So first topic will be fertilization. Second is anterior posterior polarity. Third is dorsal ventral polarity. Fourth will be segments in parasegment formation, how the segments are formed on the body of Drosophila. And last topic will be homeotic selector genes and their function. So before starting to all this, I just want to clear it. What is anterior, what is posterior, what is dorsal, and what is ventral. Though in the previous classes also I have already told what is anterior and what is posterior. So let's just remember it or just brush up what we learned in the previous class. So anterior is something towards the mouth. Here, as we can see, anterior is towards the mouth of Drosophila. Okay, and posterior is towards the tail. Posterior is towards the tail. Fine. Means if a Drosophila is lying on like this, the part or the pole towards this face, where its eyes and mouth is there will be anterior portion or anterior pole will be found over there, and towards the tail, posterior part or posterior pole will be found. What is then dorsal and what is ventral? So dorsal is the face that is facing towards the roof side here. Okay, towards the roof it's dorsal and towards the floor it's ventral. Okay, ventral means if suppose the Drosophila is there, it's lying towards the stomach, lying over the floor, and its back portion is towards the roof, then the back portion is dorsal and the part that is facing towards the floor is ventral, and the part that is facing towards the mouth is that pole will be anterior, and the pole that the part that is towards the tail of the Drosophila is posterior. So I hope this is clear, what is anterior, what is dorsal, what is ventral, what is posterior, because if we are confused in this, then it will be little difficult to understand how these polarity are formed and what are types of proteins or genes are involved in this process. So I hope you all have understood what is anterior and what other poles are. Then Drosophila, uh, development in Drosophila is a orderly sequence that is basically controlled by differential expression. What does this mean? This means that the process of development in any organism is always an orderly sequence because each thing is linked with one another in some or another manner. Okay, so development in Drosophila also is an orderly sequence that is basically controlled by differential expression. Differential expression means what? If one protein get expressed, then other protein or other genes are also linked with that, as it is already orderly sequence. So it is basically dependent on the expression of each and everything. So the if we want a proper or wild type of Drosophila, that where the development have occurred nicely or in a proper way, so in that case that is controlled by differential expression. Apart of this, the Drosophila displays a holometabolous method of development. Then what does mean of holometabolous uh, method of development? Holometabolous means any organism that have distinct stages or three stages in their whole development life cycle. First is post-embryonic life cycle. That means there are three distinct stages in their post-embryonic life cycle. First is larva stage, then pupa, and then adult, that is imago. So if any organism is containing these three stages, first is larva, then pupa, then adult, then such development is known as holometabolous. Then, as I told, adult structure is known as imago. So imago consists of head, then there are three thoracic segments, then there is the abdominal segments. So there are three thoracic segments, T1, T2 and T3. Each have their own function, or each develop to form something or some part of Drosophila. Then there are abdominal segment. Abdominal segments are 9 basically, from A1, A2, A3, A4, A5, A6, A7, A8 and A9. So as I told, there are three thoracic segments. So T1, T2 and T3. T1 develops to form pair of legs. T1 develops to form pair of legs. Okay. Then T2 forms or develops to form pair of legs plus pair of wings. Then T3 developed to form pair of legs plus pair of halters. If we remember the structure of a Drosophila, then we will know that it contains legs, then there are wings, and there is a pair of halters or halters. So the T1, the first thoracic segment, forms the pair of legs. Second thoracic segment forms pair of legs plus pair of wings, and third thoracic segment form pair of legs plus pair of halters. So this is the condition of a wild type. When there is no mutation, Drosophila is of wild type, then these segments will form these functions as I told. If there is a mutation case, if any mutation has occurred, then there will be changes, and what changes, that I will deal in the last part of the topics that I told to deliver. Last topic is homeotic selector genes and their function. So in that case, in that class, I will tell what will happen if mutation occurs. Then the first topic of this whole Drosophila developmental biology part is fertilization. So the point is how the embryo is formed, and the process of formation of embryo is known as embryogenesis. So embryogenesis begin at fertilization. Okay, means once a fertilization is done, then embryogenesis, the formation of embryo starts. But in case of Drosophila, the main or very important or critical events occur during oogenesis. Oogenesis is what? Oogenesis, as we all are aware, oogenesis is formation of oogonia, of female germ cell is formed in this process, that is oogonium. So this whole process where the female germ cell, the oogonium, is formed is known as oogenesis. So Drosophila means the embryogenesis starts at that stage only. Each oocyte is descended from a single female germ cell known as oogonium. Okay, so if a oogonium divides 4 times, means a single oogonium, when it divides 1 time, then it forms 4 cells, 4 interconnected cells. Fine. If it divides 4 times, then it will produce 16 interconnected cell, in which one will become oocyte and other 15 or remaining 15 will form nurse cell. Okay. And this whole cluster of 16 interconnected cell is surrounded by ovarian follicle cells. Here, as we can see, the surrounding our ovarian follicle cells are present that forms a egg chamber. Means inside the egg chamber there is one oocyte and then 15 nurse cell, and all this cluster is surrounded by ovarian follicle cells. Together they form egg chamber. So what is the difference between that oocyte, the follicles that are surrounding the oocyte, and the follicles those are surrounding the nurse cell? What is the difference? So basic difference is that the follicles that are surrounding this oocyte, as here we can see, the like follicle cell that are surrounding this oocyte, the morphology of those follicle cell is columnar in shape. Okay, this is columnar. Fine. Whereas nurse cell is surrounded by squamous follicle cell. Here this follicle cells that are surrounding this nurse cell is squamous follicle cells. Fine. Then nurse cell provide mRNA and protein to the oocyte. So what is the function of nurse cell? As we know, 15 are nurse cell, 1 is oocyte. Till this it's clear. Then what is the function? What is the purpose of forming this nurse cell? Nurse cell basically provide mRNAs and protein to the oocyte, means it somehow give nourishment to the oocyte, and this nourishment is in form of mRNAs and protein. Fine. And the anterior posterior polarity of the embryo is established while oocyte is still in the egg chamber. Means when oocyte is still in the egg chamber, anterior posterior polarity of this embryo is established. What will be the anterior part, what will be the posterior part, it's already established in this egg chamber. Okay. Then what happens after fertilization? Once fertilization is done, fertilized egg is there, then what happens? So after fertilization, there is one nuclei here. There is one nuclei, and this nuclei goes to 9 rounds of nuclear division to produce multinucleated syncytium. Syncytium is what? Syncytium is the diploid nuclei undergoes series of karyokinesis without cytokinesis. Means here, as we can see, there was one nuclei. Okay. Then after 9 rounds of division, it formed 9, means more than, or it's now multinucleated, as we can say. So how it is multinucleated, but the cell is single. So in that case, what does this mean is there is no cytokinesis. No cytokinesis means the cell is single, it has not divided, but the nuclei has divided to form a multinucleated syncytium, and this formation, or here nuclei has divided, so this process is known as karyokinesis. And this type of structure with multinucleated, there are so many nuclei present in a single cell, so this step comes after 9 rounds of nuclear divisions that have produced multinucleated syncytium. So this we have to remember, that the here only karyokinesis occurs and there is no cytokinesis. Syncytium is, we can see here, is there is a single cell and there are many nuclei residing in the same same cytoplasm. They are sharing the whole cytoplasm present in the cell. Because cell is single, so cytoplasm is also will be common for all these nuclei. After this, what happens? Here, as we can see, nuclei then migrates from mid portion to that end. Okay, here nuclei migrates from the mid portion to the corners, where they form a syncytial blastoderm. Here it forms a blastoderm. Here, as they go towards like surface, there they form a syncytial blastoderm. Then what happens? Plasma membrane encloses each nucleus. Here, as we can see, there is a compartment for each nucleus. So plasma membrane encloses each nucleus and convert the syncytial blastoderm into a cellular blastoderm. So here, first in the third step, as we can see here, it is syncytial blastoderm that is converted to cellular blastoderm. Fine. A small subset of nuclei in extreme posterior end are segregated into pole cells, which gives to gametes in adult. Means a single subset of nuclei. Here, as we can see, there are compartments for each nuclei. So these small subset of nuclei in extreme posterior end. Here, as we can see, this is the posterior end, as I told. Like if it is a Drosophila structure, then if we consider this as a mouth, then this will be anterior portion and this will be posterior portion. Okay, so this will be anterior. This will be anterior and this will be posterior, right? So here, this side, the posterior side, it will be segregated, and when like this extreme posterior, they are segregated into pole cells, and further they will give to adults. They will give rise to gametes in adults. Okay, in case of adult Drosophila, these like small subset of nuclei will form gametes in later stages of the Drosophila life cycle. Here we can see there is a like whole process I have tried to summarize in a single figure so that it will be easier for you people to remember. So let's start with the first step. In the first step, there is a fertilized egg, there is a single nucleus, the condition is 2n, and after the 9 rounds of nuclear division, means there is no cytokinesis, only karyokinesis is happening over there. So here we can see it is now multinucleated. So this is the stage where anterior and posterior polarity starts to form. This is the stage. Okay. After this, they form a syncytial blastoderm, means they migrate towards the end. After that, in the last step, the plasma membrane will enclose each nuclei, and this side, this posterior sides, whatever the nuclei are segregating towards the posterior side, they will form gametes in the case of adult Drosophila. So this is known as cellular blastoderm. Fine. After this, protein gradient forms, and how they form, how like the proper adult embryo, how it is formed, how the thoracic segments are being formed, how abdominal segments are formed, because this is just a structure. But how these wings have been formed, legs have formed, then head is also there, eye is also there. So how they develop, what are the proteins, what are the genes are involved in this process, this is very interesting to know in Drosophila, because there are number of proteins, number of genes involved in this process, but all of them are linked in a way. So it's very interesting to study about this Drosophila. I find it very interesting. So I hope you all will also find it like interesting. So this is the process of Drosophila development. Then I just want to give the brief of the genes involved, as I am telling from the very starting of this slide that there are genes and there are proteins, number of proteins are involved in this process, but what type of genes, like what are the names of those genes? So I just want to give the brief of what kind of genes we are going to study under this topic. So if we talk about the Drosophila development or embryonic development, so there are two types of basically gene. One is maternal effect genes and other is zygote genes. Okay, so maternal effect genes means mRNA and protein that are basically from mother or of Drosophila. So those genes that have come from maternal part or mother side, those are maternal effect genes. And second is zygotic genes that are formed inside the zygote after fertilization. So if we divide the zygotic genes into two, one is segmentation gene and second is homeotic selector genes. Further, segmentation genes can also be divided in three: gap genes, pair rule genes, and segment polarity genes. Gap genes, pair rule genes, segment polarity genes. Under the homeotic selector genes, as I already told how the legs, halters, and all are formed, these will study inside the homeotic selector genes and how they function. So these are the name of genes, but inside those genes, how the mutation cases rises, if there is some kind of mutation, what will happen to this wild type, how the mutant one of the side of Drosophila will look. So these are the things that we need to know or we need to explore. We'll explore in later classes. So this is the hierarchy. As I already told, I just want to give the brief of this genes. So because we have to study them in very detail. So maternal effect genes, what are the representative genes of this maternal effect gene is bicoid, swallow, oscar, caudal, torso, trunk. These are the representative genes that are received from mother side. Then effects of mutation, what will happen if they get mutated? Then there will be very major disturbances and anterior posterior organization will not form as it should be. Second is segmentation genes. In segmentation gene, there are three types: gap genes, pair rule genes, segment polarity genes. Gap genes, they define very broad regions inside the egg. Representative genes are empty spiracles, hunchback, crouple, nerves, tailless, orthodontical. So effect of mutation will be, as I told, these, as the name suggests, gap genes. So if gap genes doesn't work in a proper way, then there will be a gap, there will be missing segments in a major region of the body. And then pair rule genes, pair rule genes, basically there are 7 segments. These are the representative genes, and if these genes are not in a proper way or not in they didn't function in a normal way that it should be, then due to the effect of mutation, the part of a pattern will be deleted in every other segment. And then segment polarity genes, after the formation of broad segments, there will be also smaller segments will also form, and there are 14 segments. These are the representative genes. Then homeotic selector genes, they basically determine the regional characteristic of the whole Drosophila. So these are the representative genes, and if these doesn't form properly, then inappropriate structure will form from a given segmental level. Means once the segment is formed, still if this homeotic gene does not function in a proper way, then an inappropriate structure of Drosophila will form. So that's all for today. Thank you everyone. Thank you for watching. Thank you for watching.